Morning, everyone. You're welcome to Bike 315. I want to believe this course will be your first. We're supposed to have been the first 300 level course, but unfortunately, I was in the class. As a matter of fact, the class is still locked. So tried my best to contact the class, but people didn't. So that I can tell you that today's class is going to hold. From class members so that the ones available should try and make it to the class. Then unfortunately, when I came, nobody is there and the class is even locked. So I felt I need to record this the introductory aspect so that while waiting for the next class, because I don't know if I will have a time this week to fix the class to at least pay for this time. So I decided to record this introduction aspect to get the time to move over it. And if you have any question, then when we come for the next class, that's next week, we can discuss with the questions you have. So I will share the link to maybe anybody I can in contact. And let that person share it with your group if you have, so that even those at home can follow us in the class. So when they resume, we can will have lots to this. If you have any question, do not hesitate to when we come to bring this class. To be raising something we discuss on. In this course by 2315, title Amino acid and protein metabolism, we are going to basically discuss is that is more or less a continuation of the previous course you must have done on amino acid and protein chemistry. So we're going to have a vision overview, a summary of what was discussed in the previous course that is related to this. And, this one serves as a continuation. And in this course, it's divided into two. I am collaboration will be taking you through this course. So my part will be the first part. In this part, we are going to discuss this content as you can see on the screen. I decided to divide it into four. 4 or 5, let me see, 4. So that we can see if we can discuss this whole content in 4 weeks time, so that the fifth one will be more or less a revision class. And after revision class, the fixed test, that's the fifth week is the revision class. Then the 6th week will be the test. So this, I have already fixed the test for this course. The 6th week of this, which will be on Monday, the 6th Monday from today. Today being the first Monday. And today we are expected to cover at least the introduction aspect, the amino acid and their sources, transamination reaction and mechanism, the amination reaction and the mechanism, type oxidative and non-oxidative, metabolism of ammonia. This will be basically what we should cover in the first week. In the second week, we look into the urea cycle, its reaction, metabolic disorder into this. And then if there is anything, we can add some from the big of carbon skeleton metabolism or degradation of amino acids. So all the functions of most common 20 amino acids found in protein will be discussed here. How these amino acids can be degraded, broken down during metabolic reactions when the need to break down or to degrade these amino acids comes. So the last one, which will be the plate of carbon skeleton of individual amino acid, is something that, like I said, we are going to see for all the 20 amino acid, even though you will have 6, 5, 3, 4 that have a similar pathway, and then we will discuss them all together. We'll see the entry of each amino acid and how that amino acid is broken down to the carbon skeleton and what the carbon skeleton is being used, which we'll discuss. I know you must have known some of those things during carbohydrate chemistry of amino acid and proteins. So this is just a continuation. So this part, the last part, will take us at least 2 weeks. So the first part, 1 week. The second, which is the urea cycle, 1 week. And then there is anything we can add as part of the page of carbon skeleton of individual amino acid. And then the page of carbon skeleton of individual amino acids will take us 2 weeks. So, but if we are luckily enough, because there is no, I will try and see that I will discuss the basic things you're supposed to know. If we're able to beat it to finish it before that time, I think it will. So I'll start with the introduction. You've already known what's proteins from the previous class. You know that proteins are just like the other biomolecules, like carbohydrates, lipids, and nucleic acid. They are all biomolecules. They are polymers of various molecules. In the case of nucleic acid, we said they are polymer of peptides. In the case of carbohydrate, we say they are polymer of monosaccharides. And then in protein, we said they are polymer of amino acids. So protein, with the exception of lipid, which we said they are not a polymer, but you can have more than 3 up to 2 or 3 fatty acids linked by a particular molecule, but rather they are particular as polymers. So in protein, we say protein is one of the most abundant organic compounds. And from the discussion you had in the previous class, they are basically classified into structural proteins and intracellular globular proteins. In structural, you talked about collagen, keratin, fibrin, etc. Actin and myosin play very important role as structural proteins. Then you have intracellular, which include the hormones, the receptor, the enzyme, the signaling proteins, etc. And you've also known, based on the function, the protein have a turnover period. The protein can be degraded, broken down if the need arises, and if the need arises, it will be produced. And after it's finished, what is produced, it can be broken down depending on the function and the nature of the protein. Most especially, some of these proteins are very labile. They can easily undergo some, they are prone to damage. So some of these enzymes, that once they are only produced, once they are produced, they did what they're supposed to do. The device also they are produced like this. So this is how proteins are being produced. Each protein, all the proteins have this turnover periods. Like the proteins mostly found in skeletal muscles, mostly the structural proteins are the ones that have a high turnover period, but there are some that have even a turnover period of a minute or 2 minutes or less than that. to be produced just within a short period of time. They are broken down. And once these proteins are broken down, they produce amino acids. Then we've already known that the amino acids found on the protein, there are at least basic 20 amino acids, and they exist in L conformation rather than the D, as we have seen in the case of carbohydrate polymers. The monosaccharides, they exist in D form, while in amino acids, we say naturally found amino acids in the protein, everything is in L form, individually, and plants, you can have some of those amino acids. We really see how the body takes care of the amino acids when you take a protein plant diets containing a protein. So these amino acids basically classified structurally in the previous classes. So one of the classification, which I think just to see one of the things is that they are classified on the basis of essentiality. Ability of the body to produce it, we are classified as non-essential, while those that can be synthesized by, cannot be synthesized by the body, rather the body will meet other need of this amino acid only from diet required to be essential, even though in between them there are what we call essential. But basically, the two classifications, essential and non-essential. So the essential comes about when the production is less or when the need is high on a certain condition. A certain condition where the need is high and the production efficiency is not enough to meet after the need. That is the condition of conditional essentiality. So basically, these are the things you, I think I can remind you, and meanwhile discussing this course, we should be able to also discuss some of these things that are related to what we already learned from the previous class. Okay, so basically starting with this class, I will start the class by defining what is amino acid pool. We said amino acid pool is the total number of all the pre-amino acids found in a biological system or in an organism that are available for activity or in metabolic process. So we have known that amino acids, once they are found in the body, they are used for certain processes. But of course, these amino acids, we know that cannot be stored by the body. Like this is an excess amino acid, just like it's known in protein, in carbohydrate, where you have an excess glucose can be stored in form of glycogen in the body, and it is known in the case of fat, excess fatty acids can be stored. Triacylglycerol can be stored in form of triacylglycerols, and in the case of protein, we don't, the body do not store amino acid, or rather protein for storage or further use. Rather, once the body meet after the need for the amino acids of the amino acids, then the excess amino acid will have to be converted for usage. So let's see, we cite that the amino acids are the total amino, despite the fact that the body uses this amino acid to synthesize proteins and some other nitrogen-containing compounds. So that, of course, the body also keeps some certain level of amino acids for so many reasons, in the static readings, emergency production, and the rest. So these amino acids are found flowing in system, cytosol, many cells, different compartments. So most of these amino acids, instead of being stored like in a various form, in a different form. For example, you have alanine, glycine, isoleucine, and you find all of them in biological system. Rather, you will find that about 50% of these amino acids were converted, where in form of a glutamate and glutamate. So any amino acid that is excess and the body doesn't need it, it can be converted to glutamate or glutamine at a certain level so that it can be stored. But if it exceeds even the level that the body can give as amino acid food, then some certain percentage need to be broken down for these other things. So, but for example, we say from this figure, we can see that an average body of a human being contains about 100 grams of amino acids. So if the amino acids are more than 100 grams, meaning they're excess, so they cannot be stored in amino acid food. Rather, the excess will rather be broken down to produce some materials. Of course, it can be used to produce energy. It can be used to produce carbohydrate. It can be used to produce fat. So basically, carbohydrate. Let's say because we discuss or fat rather. You've known the classification of fatty acids, amino acids, where you classify them based on metabolic functions. They are classified as glucogenic and ketogenic. So, of course, some of them can be converted to glucose and stored in form of glycogen, or even used as energy for glycolysis, energy production, or some ketogenic can be converted to ketone bodies or can be converted to acid. So before, this is just to I'm trying to link things that has a direct connection with the previous class so that you can easily follow up with the class. So, like I said, so this excess amino acids that are being stored in the body called amino acid pool, you have 50% of these amino acids kept in form of glutamate or glutamine. We'll see why glutamate or glutamine. What are the roles of the glutamate? What is significance is storing them in form of glutamate? And because it is not that all of them are glutamate or glutamate, rather, they are converted to glutamate and stored in form of glutamate. That's exactly what happens. So that they will exist in amino acid pool as more of glutamate and glutamate. This is based on the significant role the glutamate and glutamine plays in nitrogen metabolism, which you will shortly see some of those roles as we go into the course. Okay, so this amino acid pool, there are some processes that maintain or replenish or deplete the content. Of course, the body needs amino acids to do a lot of processes because there are processes that are continuous. I just mentioned protein breakdown, turnover. So protein can break down and the new synthesized. This is a continuous process. This one is broken down, this one is synthesized, this one is broken down. I just say that some of them can even have a turnover of less than 2 units per minute like that. So just imagine that at every point in time, you can find this process occurring in the body. So meaning that the amino acid pool is being depleted or at the same time also replenished. So let's briefly look at some of these processes based on the figure here. Let me explain some of the processes. We said it is maintained regulated by a number of processes or sources. Number one, let's say a dietary amino acids. Yes, of course, when we eat food contains a lot of amino. Depending on the source of the food, of course, it contains source of protein contain proteins. Some of this even rice, some of those we consider naturally carbohydrate. Of course, they are more of carbohydrate. What it means is they are more of carbohydrate, but you can have a significant amount of proteins inside. So when you eat a food like rice, you have some digestion, you will have some of the acids that these are acids increase them. More importantly, when you eat the proteinous food, that is food that protein of the protein or the quantum which is more of the protein, so you have a lot of amino acid in the system. So this will increase the amino acid. So another thing is the protein degradation. The protein degradation, before then, I talked about dietary amino acids. In dietary amino acid, you have, like I said, you can have some organisms that most of their food is even protein. So when it's an organism, most especially like carnivores, their food is more or less protein. So whatever they eat, it's the majorly protein. So the amino acid pool will increase drastically, and then all the amino acid will have to be used for energy production for a lot of things because there is not much carbohydrate in the fatty acid. So it is the amino acid that will be converted to glucose for energy production, and carbohydrates from glycogen. It can also convert it to fat ketone bodies for energy storage. So you have about 90% of even the energy produced is actually coming from the amino acids. Amino acids need to be broken down to produce these sources that are needed for energy production, carbohydrates as well as fat. So basically, this in the case of animals like herbivores, but like herbivores that basically eat from plant source, human included. So they have a lot of carbohydrate, they eat a lot of carbohydrates. So the food they eat mostly, unless the one they eat food that contain more of protein, it's not something that is continuous all the time. It's just one the time, like you can eat a food in a week, you can eat a food more protein which continues. So the next one is the protein degradation. I just explained how protein degradation or protein turnover is. So once the protein is broken down, it's broken down to form amino acids. So that increases the amino acid content, even though at the same time also another process of protein synthesis takes place to take out this amino acid. But nevertheless, this is a process where the amino acid pool increase. Then the next one is the synthesis of the amino acids. Of course, the body also has the metabolic machineries to synthesize the amino acids. Although this is basically all restricted to the essential amino acids, non-essential amino acids that the amino acid body can synthesize. So they also increase acid. Then we have some of these processes that causes a decrease in amino acid. What are these processes? The processes include the protein synthesis, which I've already explained. And then the second one is the synthesis of the important nitrogenous compound like protein, or pyrene, the phospholipids, myelin, phospholipids, nucleotide bases, purine and purine contain protein. When you do synthesis of nucleotide, you will see how so we use amino acids as sources of that protein. Even when you do the reaction of carbonyl phosphate synthesis, you will see where the ammonia that was used by that reaction comes from. So you also have a synthesis of carbohydrate as carbohydrate, just like I explained, most especially in carnivores where there is a lot of protein-rich diet. So it is the protein-rich diet that is converted to hydrogen. Used for energy production and the excess can be stored in the glycogen. So also the case of fat can be used to synthesize the ketogenic amino acid can be used to synthesize ketone bodies and fatty acids. And the fatty acid can be used for energy stored as the case and then we have already explained energy production in the case of synthesis of carbohydrates. So it can be synthesis of carbohydrate can be synthesized and then immediately used for energy production, or it can be synthesized and then stored for nutrients. I hope this clearly explained the amino acid pool, the sources of amino acids pool, and those process or mechanisms of depletion of amino acid pool. Please, if you have any question, do not hesitate. Do not hesitate to do this often. You can, when you resume, you are free to come and ask me when you meet in the class also, please do not hesitate. Have your hand and ask your question. So I will quickly go through one of the processes I explained that causes an increase start as the source, because basically this is the major source of the essential amino acids. So I talked about digestion, even though digestion is not included in this outline. I know while we are discussing all the metabolism of carbohydrate, metabolism of fatty acids, if it's we actually talked about the digestion aspect. So I also decided just to say one or two things about the digestion, so you can read more. This is from a lineage. So I will put the reference of most of the consultation. Majority of the consultation is lineage and such a way. So you can read some of you can see that some of these people are even from lineage or such a way. So basically, but there are some that are not out of this text. So you can still consult these two if you have anything. So what do you take in a food containing protein-rich diet when you eat protein-rich diet? We've already known that the digestion of carbohydrates starts in the mouth with the help of slightly amylases. In case of heart, lipids within digestion doesn't take place from in the mouth or in the stomach. So also the same thing in the case of proteins. The digestion of protein does not take place. Or let me see, the chemical digestion of protein has also applies to lipids. Does not take place in the mouth or does not start in the other industry. But when we look at the different types of digestion, we talked about chemical digestion, So of course, the chemical digestion involving the mastication, churning of the food, yes, mastication by the teeth, then mixing it in the mouth, what we call this process of moving of the food down to the throat, down to the stomach. It's also a process, peristalsis, that also involved in mixing the food. It's all digestion process. And when it reaches the stomach, also there is a churning of the food, mixing these chemicals to be released in the mouth. It's also part of the chemical digestion. So basically, chemical digestion and then continue up to the stomach. But when we're talking about chemical digestion of a protein started in the stomach. So when you eat a food that contains protein, that food will be broken down into pieces in the mouth and then swallowed, taken down to stomach. Once it reaches stomach, and the gastric epithelial cells sense the presence of the food, then it will secrete a hormone called gastrin. So this hormone will cause the parietal cells to secrete gastric acid, which is a hydrochloric acid, and then the chief cells to secrete epsinogen. Epsinogen is a dymogen, is an inactive form of steel. So what happened when the parietal cells secrete HCl, gastric acid, the gastric acid is meant to first of all kill any pathogenic or potential organism that can cause disease, bacteria, or something that eventually mistakenly mistaken as part of the food. Even some cells that are not supposed to be part of the food. So this helps the also care of it. Specifically in the case of protein, it helps in unwinding, unfolding the 2D structure of the protein. So it's unwinding, unfolds for easy enzymatic activity. While the pepsinogen, we know what is pepsinogen from the previous class while discussing sequencing of protein. I know while discussing sequencing of a protein, we talked about the role of those enzymes that helps in the protein, a protein a peptide bond. So what happens in the case of pepsinogen? Pepsinogen is converted to pepsin. It's activated to pepsin. And what does the pepsin do? The pepsin break down peptide bonds occurring between aromatic amino acids. I think we can remember aromatic amino acids in an alanine, tyrosine, tryptophan. So this enzyme will cut any peptide bond involved in this amino acids. So at the end, you will have the peptide or the protein producing a peptide, a lot of peptide fragments, a lot of peptide fragments. So these peptide fragments, after all the digestion, remember these things is taking place simultaneously with carbohydrate and others. So after all the digestion, it will now move out of the stomach and go into the small intestine. When it's entered the small intestine, the acidic pH in the stomach will trigger the release of pancreatic another hormone called secretin. This secretin is from the pancreas. So what does it do? It's facilitated the secretion of this pancreatic enzymes and pancreatic juice. With the pancreatic juice containing so many things, enzymes included. And some of those things include bicarbonate, which helps in maintaining, regulating the or increasing the pH to at least a neutral pH, so that it's allowed the enzymes to perform their functions. So once they are released, it's moderate the pH, moderate the pH, and then the enzymes will be released. These enzymes include trypsin and chymotrypsin as zymogen form, as trypsinogen and chymotrypsinogen, which we basically know. And I think these enzymes are very much familiar with from secondary school biology. So have known all of them. Zymogens, released in the form of the inactive form. Basically, why in inactive form? Because they can easily cause damage to these cells. So if they are to be released in form of active, or they are produced in form of active form, they can cause a lot of damage. And there is a disease, acute pancreatitis, that if these things, by mistake, something causes the blockage in the path through which these enzymes are released, and it eventually lead to activation of the enzyme within the pancreas. It can lead to inflammation of the pancreas, and the inflammation of the pancreas can lead to what we call the acute pancreatitis, which is characterized with pain, a lot of inability to digest, so many things. So this zymogen will be activated by enteropeptidases, another enzyme that will be released at the same time to do that. So you will have the active trypsin, the active chymotrypsin, which also helps in further breaking down this peptide fragments to much simpler fragments. So this fragment will also move down to the upper part of the small intestine, where the absorption, intestinal mucosa, where absorption takes place. So in that region, also more of this enzymes somewhere, it's like carboxypeptidases and aminopeptidases. Carboxypeptidases, aminopeptidases, we all know what they do, very sure. Aminopeptidases from the amino terminal and the carboxypeptidases, we keep on cleaving the amino acid from the terminal end. We have also brush border enzymes. So, you come across brush border enzymes also in digestion of carbohydrates. So the brush border enzymes like dipeptidases can also give any form of dipeptides. So, but despite that, you still have some of those dipeptides that can escape all this process. So you will have absorption that takes place at the intestinal mucosa. After absorption, you will have some of this expand system in the body, thus contributing to the health of the still is clear. Okay. Having said that, the next thing I will talk about is transamination reaction. Why transamination reaction? Why talking about these reactions? These reactions play a very important role, central role in amino acid metabolism. So that's why we decided to discuss about them separately. So by the world discussing any other either degradation, any old synthesis, then information. All this is already known what happened in it. So that we don't have to see much. So in transamination reaction, it's just an amino group transfer. Amino group is transferred from one amino acid to form a keto acid. So it's from one amino acid to another to a keto acid. So the amino acids in which the amino group is removed, it forms a keto acid. You can see L amino acids converted to keto acid. What you will receive the amino group is another keto acid, which is most cases is alpha-ketoglutarate. Alpha-ketoglutarate, once it will receive an amino acid, amino group, it's from glutamate. This is, you can see why somebody will say, okay, why glutamate? Why glutamate is simply because glutamate is one of the major source of ammonium or nitrogen in biosynthetic pathway or even in the excretion of nitrogen. So when the nitrogen is needed to be excreted, the glutamate serves as the source of this nitrogen. When the nitrogen is needed for the synthesis of a particular molecule, glutamate plays a very important role as the source. So when you have excess of this amino group, of course, you can give it to glutamate. So that glutamate, whenever there is need of it, to dispose. This is one of the reasons why we say glutamate. And glutamate also find it easy to undergo the amination reaction. We'll discuss the amination reaction. So the major pathway of the amination reaction involves the glutamate. Although there are other pathways of the amination reaction, but they are in comparison to the one involved glutamate. It's less than the other ones, which we will shortly discuss at this reaction. So how does this transamination reaction take place? The transamination reaction is catalyzed by an enzyme called aminotransferase. And these aminotransferase, almost all the amino acids have this aminotransferase. You can have alanine aminotransferase, aspartate aminotransferase. All of them have all the amino transfers that are important, transfer of that specific amino group from that specific amino acid. This is trying to get the specificity. So, what is unique about this enzyme is the presence of a prosthetic group. It has a prosthetic group called pyridoxine phosphate. This pyridoxine phosphate is a derivative of pyridoxine. And we know what is pyridoxine vitamin B6. Yes, vitamin B6. So it has this pyridoxine phosphate in the active site of this. Why is pyridoxine? Right. So in amino acid transfer reaction, you have the amino acids that contain the amino group that needs to give out the amino. Will come and enter the active site of this enzyme, and this enzyme, within the active site, there is this pyridoxine, and this pyridoxine will react with the amino group of the enzyme coming in. When it reacts, you know, in the enzyme catalyzer reaction, there are mechanisms of enzyme catalysis. So binding of enzyme or the substrate causes a kind of changes in even the morphology of the enzyme itself. So that can lead to some sequestration. So binding of this active site of this amino acid to the active site, which contain this pyridoxine phosphate, will cause a reaction between the carbonyl carbon. You can see the carbonyl carbon, if you can see my cursor here, with the amino group of the amino acids, then it leads to formation of a bond. Once the bond is formed, there is also a conformational change structurally that can weaken the bond. This bond, for example, this is not the bond that is formed. I will explain what is this, why this is like this here. That will end up breaking this bond so that the amino group is released or attached to the pyridoxine phosphate to form what we call pyridoxamine phosphate. So you will have the amino acid that comes in with the amino group removed. So before this amino acid comes into the active site, you have the pyridoxine phosphate in the active site with this carbonyl carbon. So the carbonyl carbon, it does not exist just as carbonyl carbon until when the amino acid comes in. No, there is lysine in the active site of the enzyme. And this lysine, we know lysine have the secondary amino group. So the secondary amino group, the epsilon, call it epsilon amino group, on lysine, it is the one that interacts to form this bond with the carbonyl carbon of pyridoxine phosphate. Until when, so this is how it looks like when it's formed this. So when an amino acid come into the active site, so this conformational changes will lead to the release of this amino group of lysine, and then the bond will form with that of these amino acids, and the conformational changes that will occur will lead to the cleavage of this bond. Now let's assume that this lysine is the amino acids that need to be that need to transfer the amino group. So this bond will end up broken, and once the bond is broken, it leads to formation of the pyridoxamine phosphate. So you have this amino group being attached to this carbon. Initially, this is the carbonyl carbon here. You can see it here. So once the cleavage of the amino group, leaving the amino group attached to the carbonyl carbon, you have something like this. Will this new amino group, as the amino group that comes from the amino acid, then to be, that need to transfer the amino group. So the amino acid will now be released as keto acid. Now, alpha ketoglutarate will now come into the active site, and it will also react with this, with the amino group that is attached to pyridoxine. And then this amino group will be transferred to alpha ketoglutarate, and alpha ketoglutarate will be released in the form of glutamate. This is a classical type of reaction mechanism we call ping-pong mechanism. I think if you do enzyme chemistry, you must have done reaction mechanism, sequential mechanism, where you talked about bye the rest, and you also talked about ping pong. Ping pong, you have you're dealing with two substrate. So also in sequential, you're dealing with two substrate. The question is which one, do they come all together, and which one goes out, which one follows. In the case of ping pong, you have the two substrate not coming together. Rather, one substrate will come and bind to the enzyme, converted to the product release, just as we've seen. And then the second substrate will come in, also converted to product. This is ping pong mechanism. So this enzyme aminotransferase is a typical example of ping pong mechanism. Yeah. So now you have this deamination, transamination takes place. So when you have excess of all these amino acids in the system will convert the amino acids, or it will transfer the amino group to alpha ketoglutarate, so that you have a lot of these amino acids in form of glutamate or converted to glutamate, or the amino group being transferred to alpha ketoglutarate, forming a lot of glutamate, and then the keto acids. The keto acids, depending on the nature of the amino acids, if it is glucogenic, can be used to synthesize the glucose. If it is ketogenic, can be used to synthesize fat, depending on the nature of it. So this glutamate will remain in the system. So the glutamate can undergo another reaction. Remember, we just mentioned that it can easily undergo the amination reaction. Of course, you can have some of these amino acids that can also go through the amination reaction. But this deamination pathway involving glutamate we said is the most common one. So let's now see. In the amination reaction, what happens is the removal of the amino group. Here is not the transfer of the amino group, it's the cleavage of the amino group, then released in form of ammonia or ammonia in the system. Because it exists in the form. So the amination reaction involves the removal of amino group from the amino acid, usually in a glutamate, to form a keto acid. Which keto acid now? The glutamate, the amino group of the glutamate is removed, it's from alpha ketoglutarate. And we know alpha ketoglutarate is anyway. Okay, so in most cases, this amination, deamination, and transamination takes place at the same time, simultaneously in most cases. So it is called trans-deamination, but not in all. Like I said, some amino acid does not even go through transamination; they go directly to deamination, which we will see. So, deamination reaction is classified into two. It can be oxidative and non-oxidative deamination. It is called oxidative deamination when the deamination, the process of the removal of the amino group, involve oxidation reaction. Now, it does not involve oxidation reaction. It's called non-oxidative deamination. So we'll see example of oxidative deamination, at least 2 enzymes, 3. Then we'll also see example of non-oxidative deaminations. In some of these example of amino acids that undergo this non-oxidative deamination. Number 1 is the glutamate dehydrogenase reaction. In glutamate dehydrogenase reaction, this is the pathway that involves the glutamate. So remember, a lot of amino acids are converted to glutamate. This is the major, that is why this is the major part of it. So glutamate is important. So the glutamate dehydrogenase is an enzyme that uses a reducing equivalent. Like remember, I said the glutamate can be used in synthetic and in degradative perfume. So it can use, the enzyme can use both reducing equivalent as NAD or NADH. So NADH plus H or NAD. So it can form the NAD, like we said, when NAD is involved, it's mostly in degradative phosphate, where energy is being produced. While when it's NADPH plus H, it's usually in the reductive pathway that is involved in biosynthesis, in the synthesis of amino. So this enzyme can actually use either of the two, depending on what is the glutamate, what type or what the amination in which that glutamate is involved. Is it in a biosynthetic pathway or degradative pathway? So the mechanism of this, first of all, remember we said oxidation and then deamination. Or the two can happen concurrently. So what happened in the case of glutamate dehydrogenase is you have the oxidation first. So the molecule is oxidized. How is it oxidized? It is oxidized in such a way that the two protons were removed, and a reducing equivalent in form of either NADH plus H or NADPH plus H, as we explained, that introduce a double bond on the alpha carbon or carbon 2 of the amino acids. And that leads to formation of what we call alpha aminoglutarate, which is an intermediate. Alpha aminoglutarate on hydrolysis release the amino group in form of ammonia from alpha ketoglutarate. One thing we know about alpha ketoglutarate, if we do carbohydrate metabolism, alpha ketoglutarate is metabolite of citric acid cycle, and that can also be a connection between this nitrogen metabolism and citric acid cycle. Meaning this metabolism or this reaction increase the citric acid metabolites. So if, assuming the citric acid pathway is operating at high, there is high production of energy to the point that even the energy is enough, high ATP. And this pathway is there in adding more alpha ketoglutarate. Then this pathway needs to stop. That is why high ATP, when there is high ATP in the system, either ATP or GTP, can actually inhibit this reaction. But when there is low ATP or GTP, there is high ADP or GDP. So high ADP or GDP can actually activate this pathway so that or give us more of allocated glutarate. We need more energy to see a cycle. So this is basically oxidative deamination involving glutamate hydrogenase. So ammonium is always. So you can see alpha ketoglutarate. Most of those amino acids now are somehow converted to alpha ketoglutarate, which is to citric acid cycle as the citric acid cycle. So the ammonium is being produced inside the cells, cytocells, of many cells. So we'll see in that, but we are still discussing oxidative deamination. What are the other methods, means through which oxidative deamination can take place? So oxidative deamination can also take place using, can be carried out using amino acid oxidase. Amino acid oxidase is a pliable protein containing this pliable mononucleotide. It's a monooxygenase. It's an enzyme that uses an oxygen So first of all, instead of the glutamate hydrogenase uses NADP, a reducing equivalent, to causes the oxidation. But this enzyme uses oxygen. So first of all, it uses the oxygen. First, the proton is removed from the amino acids, being transferred to the plabine mononucleotide. From the plabine mononucleotide, it is then converted to the final sector, which is oxygen, to produce hydrogen peroxide. So the hydrogen peroxide we know can be converted, because sometimes hydrogen peroxide in the system can be dangerous, can be converted then to water by an enzyme called catalyst. You get to know this reaction. I don't know in which class, but it's something that's very common. Actually, so you have this enzyme catalyst that will convert this. So the process of this oxidation follows directly with the removal of the amino. So the amino acid oxidase are two. Can be L-amino acid oxidase, can be D-amino acid oxidase. The activity of L-amino oxidase is very low because the L-amino oxidase only acts on L-amino acids, which are found in the biological system. And all of these L-amino acids were converted. The majority of them were converted to glutamate. Majority. So you see the activity of L-amino acid oxidase is very low. But for D-amino oxidase, because when you eat a food, usually protein-containing diets from the plants, they contain a lot of D-amino acids. So this D-amino acid will have to pass through this pathway to produce the ketone acids. So they follow this pathway. That is why the activity of D-amino acid is high, especially when diets from plant source. They contain high content of protein. Please, you need to know this. Put that in here, okay? Now, we have non-oxidative deamination. In the non-oxidative deamination, basically, we have two reactions. Amino acid dehydratase, which involve the Morales dehydration reaction. In the process of dehydration, which is removal of water, follows the removal of the amino group. And this is basically related to metabolism of serine, threonine, and homoserine. While you have amino acids, the sulfohydrous, usually sulfur containing, where you have the sulfur being removed as hydrogen sulfide, and at the same time, the amino group. So, in all these reactions, we have the removal of amino group in form of ammonia. So this ammonia, we know, because the reaction takes place in almost all the organs, all the organs and all the cytosol, so you have high amounts of ammonia. So of course, this ammonia can be moved to the blood, and once the level of ammonia in the blood is high, can lead to hyperammonia. So hyperammonia, hyperammonemia can lead to toxicity of central nervous system. It's very toxic to central nervous system. We will see how it is or why it is toxic to central nervous system. Now, so all these ammonia need to be transported to the liver. When you get to the liver, then the liver will determine, depending on it, need to be excreted, or it can be diverted for the synthesis of protein. So how are they transported? There are basically two vehicles, two means through which this ammonia will be transported. Number one is the glutamine, and it is transported in form of glutamine. This is in most of the tissues. The ammonia produced by glutamate hydrogenase, or even other process, other deamination process is being transported from glutamine, or in the case of muscles, is transported in form of alanine. We will see how this transportation takes place. Now, metabolism. Now, in other tissues, you have all this ammonia. How is it transported? They are transported in this. You have your ammonia. So what happens is glutamate will react with ammonia to form glutamate. But how does the reaction take place? First of all, the glutamate needs to be activated to react with the ammonia. And the activation involves reacting with energy in form of ATP by the enzyme called glutamine synthesis. So glutamine synthesis somehow activates glutamate. How does it activate? By attaching a phosphate group to the secondary carboxylic group of the glutamate to form gamma glutamine phosphates. So gamma glutamine phosphates now have the sufficient energy to reactivate ammonia. So this same enzyme, glutamine synthesis, will now combat or will now catalyze the reaction between the ammonium and this activated gamma glutamine phosphate to form L-glutamine. So once L-glutamine is formed from all the tissues with the exceptions of muscles tissue, the glutamine can be transported down to the liver. In the liver, in the mitochondria of the liver, into the undergo reaction by another enzyme called glutaminase, where the ammonium is released. Therefore, a glutamate is formed. This is how ammonium is being transported from all the tissues with the exception of muscle tissue. And in the muscle tissue, what happened? In the muscle tissue, the transportation takes place in the form of what we call the glucose-alanine cycle. I think a glucose-alanine cycle, you did something called Cori cycle. Cori cycle, you talked about transportation of lactate from the muscles, skeletal muscles to the liver, where gluconeogenesis takes place. During when we are talking about extraneous exercise, and the skeletal muscles are highly active. The major process, major mechanism that gives energy to these muscles is usually anabolic, where glucose is being broken down to pyruvate. So you have a high concentration of pyruvate and lactate skeletal muscles. So also, that energy is needed also. Some of the tissue protein can be broken down to produce this ketoacids. So you have a lot of transamination reaction to form ketoacids from the amino acid released from the tissue degradation of the skeletal muscles. So these amino acids converted to ketoacids for energy production, and then the glutamate formed from the transamination reaction can actually undergo another reaction where you have a glutamate, because we have a high glutamate, we have a pyruvic acid. So glutamate can react in the muscles. Glutamate can actually react with pyruvate by alanine aminotransferase, so that the amino group will be transferred from glutamate to pyruvate to form what's alanine. So alanine can be transported. We know alanine is amino acid. Alanine can be transported, serve as a vehicle for the transportation of ammonium from muscles into the blood and then into the liver. In the liver, the alanine can be converted back by the same enzyme, alanine aminotransferase, to produce pyruvate and then the glutamate. The glutamate, of course, can undergo oxidative deamination inside the mitochondria, and then the ammonium is released. What will happen to ammonium, we'll talk about shortly. So while for the pyruvate, once it's transported to pyruvate to the liver, in the liver, pyruvate can be used to synthesize glucose through gluconeogenesis, which I think you want to see. This is basically glucose-alanine cycle. So please take your time, go through, understand how the reaction takes place. You know, this is, although I don't need to show the chemistry of the reactions here, you all know it's transamination reaction, which is already shown. This is gluconeogenesis, which we've discussed in the previous class. So then the other ones are just transportation. Conversion of alanine to ammonium, we talked about the amination. So it's already, the only thing we see is the page of ammonium inside the mitochondria of the liver. But before then, let's talk about the toxicity of why ammonium is considered toxic. Remember, we just said that the toxicity of ammonium is actually been reported to be toxic to central nervous system in the brain cells. How is it? Because it is associated with the depletion of neurotransmitters, gamma aminobutyric, and as well as it causes the change in the cellular osmotic balance. It also causes a depletion in energy production. That leads to coma. So one of the main symptoms is the onset of comatose, which basically has to do with the reduction in energy. Right? How does it reduce energy? Or how does it cause changes in osmotic balance? It causes changes in osmotic balance, also related to what we call cerebral edema. You know what is cerebral edema? Increase in the water uptake in the brain itself, so that you have accumulation of water. That can even lead to crying out, pressure, tension out, crying. So, how does it happen? When you have high blood level of ammonium, this ammonium can be actually transported to the brain, pass through the brain, blood brain. Get to the brain. What happened? So many things can happen. So many things can happen. Number one, you can have the metabolites of citric acid cycle being converted to alpha-ketoglutarate, being converted to glutamate. So that will reduce the citric acid cycle. Among the citric acid cycle, general energy production rates. So general energy production will affect it. That's number one. Number two, so this causes the low energy production. The second one is that the ammonium can, we know ammonium can react with the glutamate by the glutamate synthesis reaction. So glutamate synthesis reaction requires energy. So high amount of energy will also be consumed. And that also reduces the energy. So, and also, because the glutamate is also a source for this reaction, because a lot of this ammonium that goes there will be converted to glutamate. So even the available glutamate will be converted to glutamate. And one important function of the glutamate in the brain is serve as the source of this gamma aminobutyric, which is the neurotransmitter. So if this is converted to glutamate, then the glutamate is not available for the production of gamma aminobutyric. And that will also lead to problem with neurotransmission. And the third one is we have a high accumulation of glutamine. And one thing about the glutamine, in the brain, glutamine function as osmolytes. It's housed in the accumulation of water uptake and the source. So that can lead to accumulation of water, which and these are all characterized with these conditions: onset of comatose and cerebral edema. The cerebral edema is the one that usually leads to cry. So this is basically the toxicity of ammonium. If you have any question on that, please read, understand the process. Then just ask questions. I ask you to ask questions. Okay, so what happened to ammonium? After all the ammonia produced inside the mitochondria in the liver, what happened to it? So it depends on the organism. Some organisms, like aquatic species, can excrete ammonia, a nitrogen, ammonia, amino nitrogen in form of ammonia. They are called telic. And then you have some animals, like some terrestrial animals, although birds are reptiles, birds, they're still terrestrial animals. So terrestrial animals like birds are reptiles. They excrete it in form of uric acid. They call it uric. While human, other terrestrial animals, which are the majority of the terrestrial animals, with the exception of birds and reptiles, they excrete, including humans, they excrete it in form of urea. They are called uric. Now, the next thing is, we'll see how urea is formed from this ammonia. This takes us to the synthesis of urea. So I don't know, I think I will stop the recording here, or what I will do, I will stop this one for the introduction. This will be the second class. So I will make a new recording, different recording for this class, so that eventually when I come, you can still go through this. When I come for the next class, you already have the recording for the class, so you can maybe discuss just a little and then move to the next, so that this can even help us to facilitate the process the class will find it. So meanwhile, you are here before next week, please go through the class that I just finished, and then also this one that we're supposed to discuss in the next class. So if eventually gotten all what we need to discuss, then the next class will be easier for us to just briefly go over it to the next one. Okay. For this one, we can stop the recording and then record it.